My topic is the ethical improvements to cell and gene therapy clinical trials. So precise gene editing technologies like CRISPR-Cas9 are now being used to develop clinical therapies. These therapies, dubbed cell and gene therapies or CGTs, offer unique opportunities to cure rare and previously incurable diseases. By modifying the DNA of specific cells, CGTs can resolve the genetic mutations that cause the diseases in the first place. Many ethical dilemmas arise during the clinical trial process for CGTs. Some of these challenges are new, and some have already been identified as issues in the clinical trial process, but CGTs shed new light on the issues. They impede on every bioethical principle, including autonomy, beneficence, and justice, and they must be resolved to ensure that CGT clinical trials are conducted ethically. Challenges to patient autonomy have always come up in clinical trials with regard to future biomedical research consent. Often, patients involved in clinical trials will be asked to consent to the use of their biological materials, which are obtained over the course of the clinical trials, in future biomedical research, which has yet to be determined. The ambiguity of the future research has led to questions regarding informed consent. Previously, these concerns could be bypassed by anonymizing the samples, which disconnects the samples from their original owners, but that's not possible with samples from CGT trials. These samples have had their genetic sequences analyzed multiple times, and the resulting genetic code cannot be separated from the patient that the code originated from. Patients need to be offered the opportunity to provide informed consent for the use of their samples in this future research. In short, a re-consent system or a dynamic consent system. So these systems have been proposed before, but they were met with resistance. The counterarguments have been summarized by Kristen Steinsbeck, and she states, in a dynamic consent model, participants will be asked for consent continuously, simply because each new project is a new project. Thus, they will be asked to reconsent both for trivial and essential reasons, but often the former. She elaborates that requesting consent for trivial reasons wastes time and resources and is therefore unnecessary. More recent research on the topic has demonstrated that electronic medical record systems have alleviated much of the time and cost concerns, and that the framework for requesting reconsent is already established at trial sites. Also, Steinsbeck's argument does not address the anonymity issue involved in CGT trials. A reconsent system would not be hard to implement, and in the case of CGT trials, it's necessary for patients to be able to exercise their autonomy over materials that are intrinsically linked to them. CGT clinical trials also come with safety concerns, which could lead to distrust of the therapeutics technology, much like in the case of mRNA vaccines due to COVID-19 vaccine trials. Safety concerns have arisen regarding trial duration and target disease. Cases like Bluebird Bio's sickle cell disease targeting CGT and Verve Therapeutics' heterozygous familial hypercholesterolemia targeting CGT have created safety concerns for CGTs all around for various reasons. In the case of one of Bluebird Bio's clinical trials for a sickle cell disease targeting CGT, 5 years after the therapy was administered, one patient developed cancer and another patient developed cancer precursors. The therapy fell under intense scrutiny, and all of Bluebird Bio's ongoing CGT trials were halted. After this occurrence, the FDA began implementing the current mandatory 15-year observational period for all CGT clinical trials. I propose that this observational period should be converted into the mandatory trial length for rare and/or incurable disease CGT trials. Cancer trials are already about 13 years long on average, so adding 2 more years isn't that much longer, especially when considering that these technologies are very new in clinical practices. Also, changing the observational period into trial length would help improve patient participation over the entire trial duration and would increase public trust of these novel therapies. In the case of Verve Therapeutics' heterozygous familial hypercholesterolemia targeting CGT clinical trial, the trial demonstrated that perhaps CGTs should not be used to treat non-rare diseases just yet. During the phase 1 trial, 2 out of 10 patients experienced serious heart attacks, one of whom died. This trial was designed to take place over 2 years, which is a year shorter than the average cardiovascular disease clinical trial length. Extending this trial to a 15-year length would probably be excessive, but the question remains as to how to appropriately determine a clinical trial length. Traditionally, clinical trial lengths have been based on the diseases that they treat, but CGTs utilize new technologies that require changes to be made to established practices. The risks associated with CGTs outweigh the potential benefits when an alternative is available. Michael Torres, the co-founder of a CGT company and a cancer biologist, states that the sentiment for editing when there are viable alternatives is going to be a challenge. Lastly, CGTs pose interesting challenges to the bioethical principle of justice. Because CGTs seek to cure genetic diseases, they are often isolated to particular demographics. As such, clinical trials should be conducted in areas where the most people suffer from the target diseases. Challenges arise when that location is somewhere without CGT-capable medical infrastructure, such as low and middle-income countries. For example, the majority of people that suffer from sickle cell disease are located in sub-Saharan Africa, but many of the countries in that location lack the resources and infrastructure to safely administer CGTs. Further, conducting trials in foreign countries comes with concerns of medical imperialism. To conduct trials where the trials are needed, the most, and to avoid allegations of medical imperialism, pharmaceutical company sponsors of CGT trials must work to develop the medical infrastructure that is already in the destination location. One such model for doing so is the hub and spoke model. In the hub and spoke model, the hubs are healthcare or medical academic facilities with experience and capability to administer CGTs, and the hubs are then supported by spokes, which are secondary facilities with minimal CGT experience, but can provide limited services that complement the functions of the hub. Lastly, partner spokes support spokes in a similar way that the spokes support the hubs. Funding and resources go first to the hubs, then the spokes, and then finally to the partner spokes. Models such as this clarify how to best allocate funding and resources so that as many patients as possible in low and middle income countries can receive CGTs. So overall, the recommendations to appease the bioethical concerns in regard to autonomy is to adopt a reconsent or a dynamic consent system. With regard to beneficence, is to focus CGT development on rare and or incurable diseases and to convert the mandatory observational period length into a mandatory trial length. And lastly, for justice, to encourage pharmaceutical company sponsors of CGT trials to adopt infrastructure development models to ethically conduct CGT trials in low and middle income countries. Any questions?
Great, very well done, thank you. The floor is open. Questions, comments? Cece.
Lynn, thanks. That was a really interesting presentation. I appreciate all the different aspects you brought forward. I'm curious your thoughts around the potential for conflict of interest if we begin to expect sponsors to fund healthcare systems in areas, especially in LMICs, where there might be insufficient healthcare systems to be able to support CGTs or other types of infusion or similar types of trials, and how that incentivization might potentially impact the principle of justice.
Yeah, absolutely. And that's a very interesting question. And you had said you work with Novartis, correct? So I know somewhat well about Novartis's efforts with the Bill and Gates Melinda Foundation to establish sickle cell disease treating centers in sub-Saharan Africa. And that the Bill and Melinda Gates Foundation is helping to fund the advancement of the CGT treatment in general. And I think models like that, where you have either nonprofit organizations or even governmentally sponsored organizations that would be able to provide a lot of the financial resources to the pharmaceutical company sponsors that both help the sponsors develop the drug and create the infrastructure developing models in those LMICs would definitely be easier. Of course, that also still comes with even more concern of medical imperialism, but I think the concerns in medical imperialism are never going to go away. It's just they're much easier to appease or at least be wary of when we're developing the actual infrastructure that's present in the LMICs, as opposed to bringing our own infrastructure there only to take it away after the trial is conducted.
Thank you.
Barbara, did you have your hand up?
Yeah, I just this is focused on somatic cells primarily.
Yeah. So
And-
currently there's
yeah.
the global moratorium on editing germline cells. Really, we're only ethically and legally allowed to edit somatic cells.
Yes, no, so I wondered though, did some of these recommendations you were thinking of once things progress, which they almost certainly will, were you sort of playing with the idea of what would happen once we move past somatic cells, and whether this framework would be helpful, we'd need to expand it, or?
To be honest, I really hadn't considered it yet. I think our regulatory and ethical framework for the gene editing technologies and therapies that we have now is already lacking enough that we need to catch up in that regard before we even start thinking about what's going to happen when the technology advances to a point where we need to reconsider all of this again.
And I meant to say, great job. Thank you. A lot of work needs to be done, so I hope your voice is loud in this area.
Great job. I was just going to say, I was struck that I think it was Verve, the Verve company, that they would be developing such a product for hypercholesterolemia, because there are very good medications for hypercholesterolemia. That they would even choose that was surprising to me.
Yeah, absolutely. And I know actually just after I had finished submitting this paper, it came out that Verve had actually canceled their clinical trial with the current therapy and are now focusing on redeveloping their lipid nanoparticle delivery system before going ahead with clinical trials on a very similar therapy targeting the same disease.
David.
Yeah, Quinn, this was really fascinating. I was drawn in by your early point about the difficulty of shielding identity or anonymizing data because genetics is your ultimate signature. Do you consider that a closed subject, or do you think that there could be some creative solution to enable some form of anonymizing of genetic identity?
I'm not a data scientist. I wish I was. I would say, maybe there is, and hopefully, AI and machine learning can help with that. But with the number of sources that our DNA codes are stored now, especially with companies like 23andMe and private companies, let alone medical companies, storing this information and the ability for this data to be leaked, it seems unlikely that we'll ever be able to completely anonymize a genetic code.
Yeah, AI will either help solve the problem or make it worse.
Yeah.
Okay, well, that
Great
was
job.
great. Thank you.